Okay, party people, let's talk about plant transport. So today we are only going to talk about plant transport. I'm not going to go on to another chapter. So it may be a little short. That's okay, just to make it easy. So let's get started. All right, so transport in plants all has to do with particularly the movement of water and solutes from the roots of a plant up through its tissues, out the leaves. It goes out through the stomata. Water leaves that way. So at the organismal level, so the level of a tree, you could ask, how does water move up to the top of a 10-story tree? It's a long way to go, right? Yeah, it's a long way to go. So this is how it works. Water moves in through the roots, okay? And we're going to talk a little bit more in detail about how this works in just a few minutes. Up through into the xylem and the tracheids. So through the xylem, okay, and out through the stomata and the leaves. And this is mainly accomplished through evaporation, or what we call evapotranspiration. So this process of water movement, water leaving through the stomata, is evapotranspiration. And the reason that water can move this higher because of two main forces, okay, in these xylem vessels, and that is cohesion, that is the propensity of water to stick to itself. It's cohesive, it sticks to itself. And adhesion, adhesive forces, where water sticks to the sides of the xylem cells. Okay, so here we have a tree. It's a beautiful tree. I should take you to a tree and just be like, here's the tree. Maybe we'll do that later. Ooh, you could look at my Instagram. Maybe we'll do that in my Instagram today. Yeah, let's do that. So if you're not following Dr. Dodd Bio Squad on Instagram, give me a follow. Wow, I've really turned into an internet person. Anyway, okay, so if you hear anything, it's my puppy. So here's a tree. It is a sweet tree, and we're gonna look at this. Here's a cross section of the root. Well, it's not a full cross section; it's a wedge of the cross section, right? So water at the root will come in through the roots, okay, and pass through, and some of that will move into the phloem tissue, but most of it will move in through the xylem tissue. Now, this is something to bear in mind. Water moves one direction. Okay, so water will move up because of a few things that we'll talk about in a minute, but it sticks to itself, so as it's moving up, it's pulling that water up, and it also sticking to the xylem tissue as it moves up. Okay, so cohesion and adhesion. Move it up because of evapotranspiration, which changes the water potential, which we're going to talk about what that means in a minute, so don't be scared. And then carbohydrates in the phloem, they are bidirectional, so they can move both ways. Okay, all right, so we've got water, we've moved in. All right, here we is. And this is actually showing you right here, this is water, only moving up. Here we have carbohydrates, phloem tissue, bidirectional, that's that red. Okay, cool. So there is through the root, once we get higher up into the, in a tree, the bark, essentially, if you've got a tall tree, okay, the shoot tissue, not root tissue anymore. So here, pretty simple. You got water moving up. Phloem tissue could be bidirectional. Okay, cool. Now, we get up here to the beautiful leaves. This is where water, so here you can see this is xylem tissue, this is phloem, and water is moving, and it's moving out through the stomata. So stomata really have a couple of functions, right? So stomata, when they open, they take in CO2 so the plant can photosynthesize, can make food essentially, but it also is a way for the plant to lose water. So that there are considerations that you have to think about. Okay, the plant needs CO2, but you have a water loss issue, especially in certain environments. Okay, so water is going to exit through the stomata, right? And CO2 is going to come in to be photosynthesized, and that will produce carbohydrates. And those carbohydrates will move through the phloem, right? So this is just showing you the cross-section of a leaf. Here's an individual stoma, here's an individual stoma. These are guard cells, right? Right, so water moves out, CO2 moves in, and of course, oxygen also, because of photosynthesis, moves out. Okay, so to remember, stomata, photosynthesis is one very important process that they are like the guards. They bring in the CO2 and allow oxygen to leave, right? But also water balance. Stomata are very important in water balance. So those two things, okay? If I get real awkward, it's because I'm talking to a camera. Like I'm trying to pretend I'm in a classroom and it's just not possible. So I'm like, okay. So that's how water moves in through the roots. Okay. Got carbohydrates going up and down in the xylem, up. Oh, excuse me, in the phloem. Oh my gosh, the phloem only up through the xylem, and then that will move out through the stomata and the leaves. Simple. You can make it a dance, right? If you're water, pretend you're water. I could even do the dance online, guys. All right, so let's talk about water movement at the cellular level. Okay, your book, I actually have your book kind of close to me so I can tell you pages to refer to. Um, so if you want to, your book on page, we're talking about water transport at the cellular level, page 758 has the various modes of water movement, so make sure you're very familiar with that. So here we is. Here are some cells. There's sweet, and you have your plasma membrane and the cell wall, and then you have these. Basically, you have a continuum of cytoplasm. You have these junctions which are open. Those are plasmodesmata, okay, in plants. So you actually have a continuation of cytoplasm through the plant via the plasmodesmata. Okay, so at the cellular level, a couple of ways that water can move. You have active transport, which requires energy in the form of ATP, and then you have passive transport, which does not require ATP. Okay, so one very important protein in this process, aquaporin. So aquaporins are pores, water selective pores that actually allow bulk flow of water. So essentially, these were discovered because scientists were like, hey, this water, this rate of um, water movement through this plant is much faster than you would expect just from osmosis, which remember, osmosis is the movement of water down its concentration gradient. It goes from areas of higher water concentration, lower solute concentration, to areas of higher solute concentration and lower water concentration. We're going to talk more about that in a minute. But basically, they were like, why is this water moving so fast? Well, it was because of the bulk flow through aquaporins. So this allows bulk flow. That is a passive transport mechanism. Okay, cool. So you got aquaporins. That's right here. Okay, so this ion channel, so not like it is an aquaporin. Okay, you also have obviously osmosis, right, where water moves down its concentration gradient. This is showing you lots of water molecules at the top. Here's your membrane, your phospholipid bilayer. Lower solute concentration, we're moving down. Okay, basically, you could think of it like this. You're quarantined. Pretend here, right? So all this water here. This water is also wants to be with its friends, basically. It basically wants to make it where it's be with its friends. It doesn't want to do anything. I shouldn't say that. Water doesn't want to do anything. It moves it away that you have a balance on both sides of the membrane, essentially. Okay, so it moves down its concentration gradient to where you have essentially an equal number of water molecules on both sides of the membrane. So if you have fewer on one side, water will move across that membrane to that area of lower water molecule concentration. Okay, so and like I said, we'll talk more about that when we talk about solutes. Okay, so you have aquaporins, osmosis, and then you have symporters. Symporters are membrane-bound proteins. I just waved at my husband. Oh, he's getting his webcam. He has a meeting. Okay, so symporters, these are membrane-bound proteins that move two molecules at the same time. So in this example, it's showing you sucrose and protons, right? But that's basically what a symporter is, and it is a form of passive transport. And that's all you really need to know right now. You'll talk more about symporters in your other biology classes. Cell biology. Okay. Then, so you have these three forms of passive transport, right? Symporters, ion channels like aquaporins, and osmosis. And then you have active transport. All you really need to know is obviously active transport requires energy, and that's in the form of ATP, right? Okay. So those are your forms of movement, the cellular level, okay, how water and ions move. And it's important to know here down here I have ion movement. So when the soil concentration of something is lower than in the plant, that will require active transport. Okay, cool. Because it requires ATP. Okay, so let's talk about water potential. You will see this again. You will see it probably a lot. I know that when many of you will take ecology, we talk a lot about water potential there as well, because it's important to water movement in plants and whole region water balances. Transpiration matters for that, so it's important to understand how water moves through plants. Okay, so let's talk about what water potential is. So whenever we talk about something's potential, potential is basically just, well, it's right here. I do it even at the computer in class. Sometimes I'm like telling you this thing, and I'm like, wow, what's a way to put this? And I'm like, oh, I have words right there. Huh. So it's free energy. Basically, potential is the capacity of something to do work. So water potential is the capacity of water to do work. Okay, and it's measured in megapascals. Megapascals are basically just a unit of pressure. So like, trying to think of how it would correspond in terms of megapascals, like 0.2 megapascals is like 30 psi. If you're familiar with pounds per square inch, so it is often used in like tire pressure measurements. So just so you know, megapascals is what we're talking about. So when you see MPa, that's what that's referring to. So water will move from areas of high water potential, high capacity of water to do work, to lower water potential. And what's interesting, just so you know, the water potential of soil is like 0, and it gets more negative. So basically water is moving to areas of more negative water potential as it moves up a tree. And there are a few kinds of forces that make up water potential. And water movement through various media, like soil, also has other kinds of potentials, like matrix potential and gravimetric potential. We're only going to talk about 2 kinds that are important for plants. Okay, so, but I like to tell you those because the world is crazy. It has so much cool stuff in it. So this is just talking about plants. There's also soil transport through soils. It's also very cool. That's a Wikipedia hole you can fall down if you want to. Maybe not Wikipedia. Maybe a soil. I'm sure there's a soil science online textbook. Okay. I get off. You guys, sorry. Come back from soil. Come back from soil, Alan. Back from soil. Okay. Back to plants. Great. So, water potential in plants. When we're talking about water potential, I'm going to move the slide a little bit. I'm going to go over and I'll come back. So, total water potential. Total water potential, and this is 2 slides up, just so you know, I'll come back, is basically, when this is represented by psi. Psi is, this letter psi is water potential. Okay, so water potential is equal to pressure potential plus solute potential. Okay, all right, and water will move until the potential of the cell is equal to the potential of the solution. Okay, it will move until those are equal. So let's go back and talk about what each of those potentials mean. First, pressure potential. Pressure potential is turgor pressure. what is turgor pressure? You're like, whoa, turgor pressure. Turgor pressure is basically the pressure exerted when, so the pressure exerted here against the cell membrane. Basically, what's happening is water is has filled the cell, and the cell is turgid. It's firm. There it is, firm. We call that turgid. So turgor pressure is what allows plants to either be nice to stand up, or if you have low turgor pressure, they wilt. So that's all turgor pressure. That's all pressure. Okay. So turgor pressure is the same as the pressure potential. Okay. So basically, you have a force here that is the water moving and hitting, like keeping the cell membrane taut against the cell wall. So this pink here in this little example, that is the cell membrane, and the green is the cell wall. So, and now here you have pure water. So water inside the cell is keeping that membrane taut against the wall, and it's taut, turgid, okay? All right, if you do not have a membrane though, you cannot have pressure potential. So here, the solution is not contained in a membrane. It's in a beaker, but it's not in a membrane, right? That will have zero pressure potential. This is going to matter for the rest of this example in a second. So water here, right? And water is pressing against here, okay? Turgor pressure. So that's the first part of water potential. Second part is solute potential, and it deals with the concentration of solutes. So, and if you see me looking down here this whole time, this is instead of looking back at the board every now and then, I'm looking here. And it's so hard sometimes, you guys, to not just look down there because it's white. Okay, so, sorry, solute potential. So, this has to do with the concentration of solutes and how they differ in the cell versus outside the cell. Okay. So in this example here, these are sucrose molecules. Sweet. Literally. Ooh, look. Still drinking my coffee. So, sucrose molecules here. So, inside here, solutes, higher solute concentration means that there is a lower solute potential. It is more negative. Because you can think of it this way. There's a lower solute potential here and a higher solute potential here. All else being equal, don't think about turgor pressure, just thinking about solute. Water will be more like, it will move into the cell to areas of more negative solute potential because there are more solutes here, so it will move down that gradient, right? So just know that water always moves to areas of more negative potential. Okay, so higher capacity of water to do work out here because it's less negative, it is higher, and lower capacity of water to do work out here. So water moves into the cell. Cool. So remember, pressure potential, turgor pressure, plus solute potential give rise to the total water potential. Okay, and that controls movement of water on the cellular level. Okay, so this is just a nice example showing you, and this is in your book. Let me tell you what page that's on. Okay, 760. On page 7, I think that's 760 real. Or it's thereabouts if you have a different edition of the book. So here you have a plant cell. The pink is the membrane. The green is the cell wall. So you have, and just recall from before, so negative 0.7 megapascals. This is the cells. So I told you the potential of the cell, water will move until the potential of the cell is equal to the potential of the outside solution. Okay, so here is the solute potential in the cell, negative 0.7 megapascals. Okay, so that is the solute potential right here. The pressure potential, 0.5. All right, that gives you negative 2 megapascals. Okay, water would not go anywhere in this situation because the solute potential, the potential of the solution, which is just the solute potential, is negative 0.2 megapascals. Okay, so the water wouldn't go anywhere in this situation, is the long and short of that. Okay, so it's good to think about this, and practice it. Use a little whiteboard and be like, okay, if there's more solutes outside the cell, and fewer inside the cell, then water is going to move out of the cell, right? Because you're going to have higher solute potential in the cell. Okay, and then that would actually cause the cell membrane to come away from the cell wall, and that would cause the plant to wilt. Okay, so let's briefly talk about how water potential changes moving up the plant. So here in the soil, again, this is water potential in megapascals. It's 0 or near 0. And as you move up the plant, it becomes more and more negative until you have, around air, it's like negative. Free air is like negative 100 megapascals. Okay, so water is moving up the plant here. Okay, and most of this movement is because of transpiration, 90% transpiration. All right. You also have some of that water movement is because of root pressure, because you have active ion uptake in the roots, and at night, stomata, at night you don't really have as much transpiration happening, so those solutes are being taken up in the roots, and so you have higher solute concentrations. So in the day, when more water's kind of rushing in, you actually have basically a lot of pressure built up in the roots. And so, like, if you cut a plant, sometimes you'll see it keep oozing. That's because of root pressure, actually. It's not necessarily just transpiration, because if it's just oozing for days, not days, but a long time, that's usually because of root pressure. Okay, cool. And mainly, the main thing I want you to know about root pressure, and let's see, so this figure is on page 762. The main thing just to know about root pressure is it's mostly at night. And basically, it mostly occurs when transpiration rates are very low or not happening. The stomata are closed. Okay, cool. It's a way for the plant to actively take up ions, and that in turn influences water transport. Okay, so let's talk about transpiration rates. So most of the water lost to the atmosphere, most of the water taken up is lost to the atmosphere. Okay, and that transpiration rate is controlled by the opening and closing of stomata, which is basically via the inflation or deflation of the guard cells around the stomata. So these are basically like guard cells, right? And then your stomata, basically, if they are inflated, the stomata will open, and then they'll close. Okay, cool. So just remember that stomata control the rate of transpiration. Most of it is eventually lost to the atmosphere. Most of that water taken up. So let's talk about water balance in plants. What do plants do when they lose too much water, or they're limited by water in some way? So they lose too much and they can't take up enough. So a few things can happen, and then we'll finish up here. They could go dormant. So some plants will do that. Some plants, and you'll see this actually in Arkansas in August. I don't know if any of you have noticed, or like in July, where you're like, whoa, there's a lot of leaves on the ground. Plants are losing their leaves. Yeah, they're getting rid of them. So it's a water retention strategy. They're like, whoo, they're like, get rid of it, get rid of it, get rid of the stomata, get rid of the stomata, right? So they can lose leaves. This particularly are deciduous plants. Bless you, my son just sneezed. Okay. You may have, so remember we talked about trichomes, those little hairs a plant may have, or certain plants have evolved to have lots of trichomes, or a waxy cuticle. Those are both to prevent water loss. Okay. Some plants have fewer stomata. That is a life history strategy to reduce water loss. And some of them have stomata in strategic places like pits of a leaf that will reduce the amount of water loss. I do also want to point out, and I'm not going to talk a lot about it in the lecture, but it's just fun. If you want to know more about other adaptations like halophytes, plants that live in really salty environments, your book actually talks quite a bit about plants that live in salty environments. And we talk about that quite a bit in ecology. But in this class, I just want you to really be familiar with these stress responses to water limitation as opposed to also um, salt adaptations. So that is all for Monday, March 30th. And hopefully I will see some of you on our live call here in just a bit. And next time we are going to talk about animal diversity. Animals. We're starting animals. And that will be, the next chapter will be the last bit of stuff for your next test. Okay, so good luck, everyone. Enjoy your day. Bye. Let me know if you have questions. If you have questions, you can put them into the discussion group on our Schoology page, or that's probably the best place to put them. And I will answer you directly, but I will also answer those questions on Friday in our live chat, just so that you can have, a real live back and forth, because that might spur on more questions. So I will see you all soon. Take care.